I'd like to introduce the next speaker, Dr. Gregory Germino, who has been a long-time PKD researcher before his more recent role as the Deputy Director of the National Institute of Diabetes and Digestive and Kidney Diseases at the NIH. Welcome, Greg.
Thanks, Thanks, George. Great, thank you. I want to thank York for the opportunity to be here. Oops, I guess, is this on? Okay, sorry, thank you. Yeah, good, thanks. I'd like to thank York for the opportunity to be here today and, of course, to be in Toronto, which is one of the world's great cities. So today I'm going to first just, I have no conflicts to declare, and my wife, who is Terry Watnick, who was the earlier presenter, she's been a co-investigator on a number of different studies. I'm going to talk about a lot of drugs very briefly approved for treatment of PKD. So the objectives for today are three. One is to understand the opportunities and challenges to develop effective therapies for ADPKD. The second is to distinguish between treatments based on downstream pathways, as we've spoken about mostly this morning, versus addressing the root cause of the disease, and that's at the genetic level. And then learn about some recent discoveries that could result in new therapies in the future. And so this is the problem that we're facing, right? So this is from a study. Now this is 10 years old, and so it'd be a great time to see an update on this. But what was really sobering about this publication is that looking at in Europe the incident rate of renal replacement therapy for ADPKD in individuals under 50 had not changed despite the use of ACE inhibitors and all the other therapies that we've been doing for the previous decades, even though there's been some change in the other later parts of the curve. And so it'd be really great to see how this changes now in the setting of tolvaptan. We would expect and we hope and anticipate that this will be shifted somewhat rightward, but this is going to be, that'll be the ultimate test, I think, of any interventions that we make in ADPKD. So this is the goal, really, is to shift that curve all the way to the right until we're all having something else that affects us. So in terms of ADPKD, we've heard a bit from Terry and others about both the slow progression of disease, and this really provides both opportunities and challenges. The opportunity, of course, is we have a long lead time to be able to identify individuals who are at risk of having the disease, whether it's through genetic analysis, if we do it early before they have any sort of clinical presentation, or even when they start acquiring cysts as they age, and we hear about the kidney volume. So there's a lot of time to step in and make an intervention. But that also provides challenges, as we've heard, because drugs given over a long time may have side effects and complications. And so this is the balance that we all are struggling with when we think about interventions. We've heard, again, earlier in the first session about clinical variability, and so we know that there are important factors that can affect the progression of disease. The genetic locus itself, PKD1 versus PKD2 versus those atypical forms of ADPKD that we've heard about, with PKD1 being more severe. We know that the nature of mutation also affects progression of disease. Truncating being those shown on the purple bluish curve that really change the most rapidly and have the earliest onset of disease typically, versus missense changes, which is the green curve, or PKD2, which is to the right. We know there's probably genetic modifiers. PKD1 has been one of them, but there are almost certainly others that can also affect in interaction with the primary mutation that may affect the rate that you get second hits or how you respond to the second hit when you get it. And of course, the frequency and timing of second hits are important parameters, and even where the second hit occurs. You can imagine a cyst coming down, perhaps in the medulla, perhaps affecting other collecting duct systems might have more effect than something further up in the cortex. There's lots of factors that could affect this. So in terms of thinking about therapeutic strategies, there's two different approaches that are currently being pursued. One is targeting downstream pathways, and we've heard a lot about that this morning. And of course, then there are those that are trying to target the underlying gene defect. And I'm going to go briefly over some examples of both. So first, talking about target pathways. So we heard very nicely in the morning session, first session, about the ADPKD pathobiology and the consequences of loss of PKD. Shown here is actually a mouse kidney on the right. This is not a human, but as you can see, it looks just like an end-stage human ADPKD PKD kidney. So you can model the disease reasonably well in mice. And what we know, of course, are some of the pathways that are then identified as being abnormal in this disease with increased proliferation. Of course, you can't get a kidney this big by not having growth. There's been reported increase in apoptosis, secretory phenotype. How do tubes get that big? Is fluid for sure. There's an inflammatory response and in fibrosis, as you can see clearly here, and certainly in human disease. So there have been many, many different pathways, signaling pathways that have been described, and Terry had touched on earlier today, that are all abnormal or dysregulated in the context of ADPKD cystic tissue. And these are some of them. I'm sure these are not all of them, but and I'm not going to review them now because we've already heard about them. But what's important to note is that for many of these, there are already drugs out there, right, that target these, and we've heard about some of these. So these are, in green boxes, you can see are the different classes of drugs that are already approved or in development for other purposes that could and have been, in some cases, tested in PKD, either in animal models and or in humans. And so there are many, many different candidate pathways. And so the challenge, right, is figuring out which ones we want, or what's going to be the balance of, again, risk and benefit in a given individual, in a given population of individuals. And so here, this is taken from clinicaltrials.gov just last week. I went online and looked to see the status of trials. And so this is a list of some of the different interventions that have been completed and or terminated over the last couple of years. And the only one that's in red, of course, is tolvaptan, because that's the only one that has been shown and reported to be impactful and beneficial and approved for use in ADPKD. All of these other agents, the everolimus and sirolimus, again, a lot of mTOR pathway, a lot of excitement about that. But it was toxic, slowed the rate of renal volume growth, but did not affect GFR. A number of these have either been terminated because of toxicity or terminated because of lack of benefit, terminated like lixivaptan, as we heard from Dr. Torres just a few minutes ago, because it was being pushed and promoted and pursued as a candidate therapy that would target the same V2 receptor, but in a way that would not affect liver, hopefully, avoid the liver toxicity, and of course, they had the liver injury, and so they abandoned the drug. There's a number of things on the far right, as you can see, are different interventions that where the failure to see significant effects is because the studies were mostly pilot studies, like pioglitazone. So there was, in fact, some benefit, perhaps, in kidney growth in GFR, but with 30 patients and with a disease as variable as this, of course, you're not surprised to find that it didn't hit significance. Same with the metformin pilot that Terry had been involved with. So a number of different drugs that have been, and some of these have been shown to be effective in animal models, and yet when we've taken into humans, either they've been in not effective or not have just not big enough to be really tested effectively. And it does raise one of the challenges that I think we have to think as a community when we're thinking about clinical trials. We have a lot of potential targets. I'm going to talk about some more that are coming down the pike. And we have a modest number of individuals, right, that we can test these interventions in. So we're going to have to think about some different types of trial designs, perhaps, to be able to figure out which combination of drugs or what sequential therapies, how we're going to be able to sort of evaluate all the different best drugs that are coming down the pike, or that some of them are, again, in pathways that have yet not been tested in a population of individuals, which is of modest size, right? These are trials that are still in progress. So multi-tyrosine kinase, tasivitinib, metformin, which is, again, this is the IMPEDE PKD, which is a much larger scale study to test metformin, HMG-CoAct reductase, SGLT2 inhibitors. There's a study ongoing right now. And of course, there's a new one, synthetic retinoid, which is targeting, again, some of the downstream pathways. And we heard earlier from, again, from Terry and from others about some of the dietary supplement interventions that are underway. The water intake we just heard about, caloric restrictions, intermittent fasting, ketogenic diet, curcumin. These have all been things that either have been tested or are being tested, and usually, though, in very, very small data sets, study sizes, and very short-term duration. So we really don't know which, if any, of these are necessarily going to be truly effective. So thinking about, again, one other pathway, and this is some data that just came out recently, so I want to just kind of talk on it a second. And you heard from Terry earlier this morning about the CDCA, the cilia-derived cyst-activating factors. So as you heard from Terry, the studies have shown in mice by Steve Somlow's group that if you knock out PKD1, of course, you get cystic disease. If you knock out the cilia, you get cystic disease. If you do the double knockout, you get a diminution, an abrogation of cystic disease. And so that's kind of, and they have this factor they think is coming somehow from cilia signaling, which gets enhanced in the context of PKD1 loss or PKD2 loss. That's the CDCA. Well, in recent studies done by Steve Somlow's group, he has identified a transcription factor called GLIS2. Transcription factors are those things, those proteins that control the expression of lots of other genes. So he has reported that GLIS2 is normally fairly lowly expressed in normal kidney tissue, but when you knock out PKD1, GLIS2 levels go way up, and that turns on all sorts of genes downstream that lead to some of the pathophysiology that we see in the disease. And of course, if you knock out the cilia, then the GLIS2 signaling goes down. And so he's tested this in mice, and that's shown here, and this is a recent publication. He's shown that if you look in the top panel, these are controls. If you look in the middle panel and the bottom panel, these are mice induced to have PKD1 turned off. The middle panel is male kidneys. The bottom panel is female kidneys. And then they've been treated with an antisense oligonucleotide. This is a short piece of DNA that can be injected into the animal that will knock down, it will inhibit the expression of the GLIS2 gene. And as you can see here, both in the histology and then some of the markers, whether it's cyst volume or it's BUN as a marker of renal function, you can see that injection of this siRNA was able to improve the cystic disease, suggesting that this GLIS2 might be a factor that's maybe upstream a lot of those abnormal signaling pathways that I just showed you a few minutes ago. So there are a lot of challenges, though, in thinking about moving from cystic pathology to finding targets to treatments. And so one of the problems with a lot of the studies of cystic kidney is that they're often done on late-stage tissues, particularly for humans, inevitably. It's also difficult to know whether the changes that you're seeing are causing the pathology or a compensatory response. So not everything that's up that you see elevated necessarily is actually driving the disease. We've actually shown in one of our studies that was a factor that was up in cystic tissue. We knocked it down and it made the disease worse. So it in fact was a protective pathway. So you can't always predict just based on the nature of whether it's up or down. And so again, and the other thing to just comment on in tissue studies is that are very rarely done to acknowledge the complexity of kidney tissue, which is heterogeneous, right? We know there's cortex, but there's proximal tubule, there's loop of Henle, there's distal tubule, collecting duct. The composition changes when you have cysts in the kidney, and the relative proportion of tissue of how many cells come from the proximal versus the collecting duct, that changes. And when you just grind it all up and you do measurements, it can be a little bit, it can give you bias. The model systems are also imperfect. The mice are pretty good, as I showed you. They get really good cystic disease. But well, how does mouse time convert to human time? If I stop a mouse from getting cystic disease for 6 months, that's great for a mouse. It's a quarter of their life. If I do that for 6 months impact on a human, is that, how big a difference is that? And so we don't really have a good way of knowing how that correlates. The kinetics and pattern of gene activation in our animal models is very different. And that may have implications in terms of how again, consequences of an activation. And the variability of disease in our animal models is not addressed. If you look at many of the animal studies, preclinical studies, they're done with very small sets of animals, 5, 10%. And yet if you look at the variability, even with a known genetic way of turning the gene off, the variability is very, very large in terms of litter mates, in terms of disease. And so these are factors that are often not completely controlled. And then, of course, as I mentioned before, the side effects of drugs are often limiting. And so the final thing, right, is we're still, even if we have everything right, let's say GLIS2 inhibition is the next big sort of downstream factor we're going to turn off and help slow the disease. We're treating the consequence of the disease. We're still really not treating the cause or underlying problem. And we're not preventing the cysts. I showed you the animals, they got less cystic, but they didn't not get not cystic. And we don't know, again, that delay in mice, what that's going to convert to in humans. We saw the data from Dr. Torres. We're going to maybe be able to push, end-stage kidney disease back by a decade in people with PKD1 or PKD2 disease. And that's great. But, if you're going from 60 to 70, you're going to be 70 and you still have end-stage kidney disease. We still have a problem, right? You're still going to be sick. And so can we do something even more effective? And that's where we really think about what can we do about fixing the gene itself? Or how can we, and there's a couple of different ways you can think about that. One is if you know exactly what it does, even if you can't get it back in there, if you know exactly what it does, you can replace that activity. It might be able to turn everything else down and turn it off, right? The other thing is what if it's just broken a little bit? Can you improve its function? And I'll give you some examples of that in a second. Can we just replace the mutant gene and protein, with a gene therapy approach? And finally, can you actually correct the gene mutation? And there are technologies that are coming, that are evolving, that allow us to do virtually all of these things. So I'm going to start with replacing its function. So this is, you've seen from Terry, this is the PKD1, PKD2 complex. The arrows indicate different portions of PC1 and PC2. PC1, that N terminus gets pushed off into the outside of the cell. The C terminus gets clipped, as you heard from Terry, and goes off to the mitochondria and maybe to the nucleus where it has other activities. PC2 has some sort of channel activity that's ill-defined. And they go to all different parts of the cell. So they've been described to be in the primary cilium. They're at cell-cell contact points. As I told you, the CTT, the C terminus of PC1, may go to the mitochondria. And they have also been described as being at where the cell contacts the basement membrane. So which function are we going to replace to be most effective? And the problem is we don't really know what it does in most of these locations, or any of these locations. So you heard from Terry that the C-terminus part of the polycystin 1 protein gets clipped off and goes to the mitochondria. And we have shown in some studies that we did with cultured cells that, in fact, if you give that back to a mutant cell, it can fix some of the mitochondria abnormalities. So we might be able to fix the mitochondria abnormalities. And so that would suggest maybe we can reproduce part of the function with that little piece. And in fact, a study done by Michael Kaplan and his colleagues up at Yale, they actually made a complicated mouse model where they turned off PKD1, and at the time they turned on this, again, a little transgene that is expressing just that small 200 amino acid portion of polycystin 1. And what you can see here on the picture on the left is the histology of the effect that this had in protecting, or at least reducing, the severity of disease. And on the right are showing just some of the other parameters that were measured in these animals when they turned the gene on. And so again, if you look at the far left of each of these B, C, and D panels, you can see that with just the gene turned off, you have a lot of cystic disease. And you can see also the variability that I was talking about there. And then if you look in the middle panel, it's showing then the BUN. And finally on the right is the creatinine. And in the middle of each panel is where they were rescued by turning on just that small little piece of the gene. And you can see it seemed to significantly lessen the severity of disease with respect to both kidney body weight and renal function. So there are studies now looking at that and testing that. That was the first report. That was a couple of years ago. We haven't heard any follow-up. I don't know of anybody that has yet repeated that study to confirm it, and so we're still waiting to see if this can work. So what about improving polycystin or PKD gene function? So there's a couple of different strategies that are being that are being pursued. So one is, can you actually boost the expression of the gene? So not only, maybe you're at this level normally, and let's say you're in again, we heard there's a 2-hit disease, so if one hit is gone, so you're down at this level, can we make that level go higher, or can we somehow improve the level of the mutant one if it's just a little bit dysfunctional? And the second is, can we improve the mutant PKD protein function? What I mean by that, I'll just show in a second. So what do I mean by boosting PKD protein expression? Here is an example. So this is just showing the normal DNA pattern. Of course, DNA gets transcribed to make an RNA molecule, and that gives rise to the proteins. This is the standard process that happens, right? So it turns out for both the PKD1 and PKD2 genes, there is, in the end of the gene, on the end of the transcript, some base pairs, some nucleotides, where there's a microRNA that's normally made and under different control that binds to this PKD1 and PKD2 transcript. And by doing so, it inhibits ability of that messenger RNA to get made into protein. So the normal mRNA gets degraded. And what happens is it keeps PKD1 protein levels and PKD2 protein levels low. Normally, they're quite low. So this is a problem. This is okay normally. This is how it works normally. But the problem is, in a state where one copy may be mutant and you want a little bit more activity from the other one, you would want to know if you can take advantage of inhibiting this inhibitor to boost activity. And that's, in fact, what has been done. So in PKD, the problem is that this inhibitor, this MIR17 that I'm talking about, that is upregulated in cystic tissue. And the reason they think it's upregulated is because I think it's under the control of c-MYC. And c-MYC is one of those factors. It's a protein that's been reported upregulated in cystic tissue for decades. It's been known about for decades. And so c-MYC goes up. That increases this MIR17, which is this microRNA, and that microRNA goes and helps to inhibit even the normal copy of PKD1. So it's even more inhibited in terms of its expression level. So the idea is, if you can block that MIR17, can you boost activity? And so here's the cartoon of that. So here's a drug, a small molecule, that binds to the MIR17, and the idea is you will increase PKD1 and PKD2 protein levels, okay? So in fact, a group, unfortunately, I apologize, the formatting got botched in the transfer from a Mac to a PC. What this was supposed to show is a paper from Vishal Patel at UT Southwestern, whereas in fact that's exactly what he's done. He has worked with a company, Regulus, to develop a small molecule that binds to this MIR17, blocks it from blocking PKD1 and PKD2, and they've shown in an animal model that they can actually improve PKD activity and increase PKD1 and PKD2 protein levels. And you can see here on the far right, they did it in some in vitro studies to show that they can reduce cyst growth. And you can see that the protein levels, as shown here for PC2, go up, right, on the far right of the panel. You can see when they're treated, when they knock out this MIR7, they can do it genetically by knocking it out, by taking deleting the sequence, or in fact by giving an injection of a molecule that again does the same thing. They were able to boost the activity of polycystin 1 and polycystin 2 and improve potentially the effects and reduce the cystic growth. And right now, there is actually a clinical trial in progress that's testing this intervention. They just did a phase 1 study that they reported where they were able to show that at least in a phase 1 study, giving this molecule to individuals, they could show an increase in the amount of polycystin 1 that's expressed that's secreted in the urine. So at least proof of principle that it might be able to do something. So boosting expression is one. Can we improve mutant PKD protein function? And so what I mean by that is shown here. In the normal state, polycystin 1 and polycystin 2 are synthesized in the ER. They are processed in the Golgi apparatus, and then they process, and then after they've been processed with some changes to the protein structure, some things added on, carbohydrates that are added to it, it then traffics to the primary cilium and perhaps to other places after that. So that's the normal process. It's been shown that in some of the missense changes, the missense change results in a form of polycystin 1 that still may be made, but it's just less functional, right? It doesn't quite have the same, it may not get processed properly. And as I'm showing here in this cartoon, you'll see this mutant PC1 is a little bit wrinkled, right? So it's not folding properly. It's supposed to go to the Golgi and get folded. It doesn't get folded properly, and it gets degraded. So the amount of functional polycystin 1 and polycystin 2 that you get to the primary cilium or wherever else is reduced. And that's enough to trigger cyst formation. So if you had a way of getting that mutant polycystin 1 that's crinkled to be more folded better, so it can actually leave the Golgi and get to the primary cilium, you would hopefully be able to boost activity. And in fact, that's what this is what's been done for cystic fibrosis, for instance. The most effective therapies for cystic fibrosis are able to take the mutant the cystic fibrosis gene, which generally results in a protein that's not very, that's less functional and doesn't go the right place. They give these little drugs, and it allows cystic fibrosis protein to get to the right place in the cell. And so the idea is the same here in PKD. Can we give a small molecule that will correct the folding of PKD1 and allow it to get to the primary cilium? And in fact, Vertex, who has some of the compounds for cystic fibrosis, is in fact now got I think a phase 1 study in progress testing these corrector molecules for PKD. Now, the one thing about this, just to say, is that so it's great. There are some drugs that might be able to work with a number of class of patients who have missense changes, but it's important to remember that the individuals with missense changes in general have less severe disease. So the ones who really need, the most effective therapy are those that are going to have truncating changes. That's where the protein either is not made at all, or it's made and it's missing large segments of the rest of the protein for function. And therefore, just adding a corrector molecule is not going to help. And so this may help with some individuals, but it's not going to help for a lot of our patients who have most severe disease. So just comment on two other things that I heard at the ASN that are interesting, I don't have on the slides for, but I heard them in Kidney Week 3 weeks ago, and I just want to comment on them because they're in the same sort of line of approach. And one of them is that a group at Yale, Whitney Bess at Yale, had identified in the upstream sequence of PKD1 a sequence that it gets translated into a mini peptide. We have no idea what it does. But what's been shown in this PKD1 and in other genes that have sequence like this is it results in a decrease in the amount of PKD1 that gets made downstream of it, and normal PKD1. And so what it's doing functionally, God only knows. But the thing is, you can manipulate this by suppressing that little piece of the gene, and you can result in boosting the PKD1 level protein. So it might be another way for people who have missense changes that have hypomorphic or hypofunctional PKD1 to be able to boost their activity. And this could be potentially effective in polycystic liver disease, where the problem is not in the PKD1 gene often. If you have primary polycystic liver disease, the mutation is in other genes that are important for polycystin 1 processing. So if you have a way of boosting polycystin 1 activity, that might be a useful strategy. And the second thing that people are thinking about, and this is not just for PKD, this is for other diseases, is if you have what's called a stop codon. So that's one of these truncating changes where you have an ATG, you have a TAG or whatever the three trinucleotide, the three base pair sequence that encodes a stop codon instead of an amino acid. There's a process normally in biology where when the translation machinery hits this, It sees this stop codon, it stops, and often the message gets decayed. It just doesn't even get made. So there are compounds that have been developed that suppress that system and allow the full transcript to be made. And so you could theoretically read through the stop codon and make a protein which might even be functional. And so there are people testing whether they can do that in PKD as well. So moving on to the last part, which is can we replace the third to last part, which can we second to last part? Can we replace the mutant gene and protein? And that means really doing gene therapy. So the idea is we have a cell that's got a non-functioning gene. We want to get the functioning gene back in and then recovers function. So you have to know anything about what the protein does. You just have to know how to get it back inside the cell. So that's the idea. And so there's some evidence to suggest this could work, even in PKD in humans. And the evidence comes from studies done by Steve Somlo's lab, where they turned the PKD gene off. They allowed them to get cystic, and then they turned the gene back on. And what they found is they actually could reverse the disease. So shown here in red is if you don't turn the PKD gene back on, you can see what happens with cystic disease. Cysts get larger and larger as the red curve goes up. But if they turned the gene back on, what you could see is all of a sudden it declines. And so if you do it early enough, before there's a lot of fibrosis, you can actually preserve function. And so in humans, right, I mean, you could theoretically even do it to people who have already some cystic disease. If you could get the gene back into them, you could stop the disease and maybe even reverse some of the cystic change. So so there's evidence to suggest this is possible. Just some more examples of that. He did it both in PKD1 and in PKD2, and in both models. Turning the gene on, the earlier you can turn it back on, of course, the better the result, the less fibrosis you have in response. But it could be effective. And again, even in late onset disease, so he waited a bit. So the disease got pretty far along, and then he turned it on and still showed benefit. And that's just showing some histology there. Okay, so why can't we just do it? It seems like a great idea. Well, so the first problem is with PKD1, the gene is just way too large to be able to use any of the conventional mechanisms of delivering genes into cells and tissues. PKD2 could probably fit, but not PKD1. And if the CTT story, though, right, that little bit of the polycystin 1 that's on the end, if that turns out to be true, that really does work, that would certainly fit within a gene vector, and that would be then an approach to therapy. But the second problem is that targeting the kidney and biliary epithelial cells is not so trivial, right, with most of the conventional methods. Now, there has, again, been progress made in this field, as I understand. I'm talking with a gene therapy guy on Monday, actually, who has been working in this space. And they've discovered a bunch of different viral delivery systems that may have affinity for the kidney, because we need to be able to get it specifically into kidney cells. And so this is, again, a field that is open and active investigation. The other challenge is getting the correct expression of the gene. You want to make sure you get enough of it, but you don't want to have necessarily too much. We don't know what the effects of having a really long-term high level of expression of PKD1 or 2 is. There's a lot of mechanisms naturally in place to keep that expression pretty low, so there's presumably a reason for that. And so what that is, we don't know. So this is one of the challenges, unless you have the native, endogenous promoter regulating the expression of this. And then, of course, finally, making sure that your gene therapy stays working over the duration of the lifespan of an individual, because sometimes these do get suppressed or get silenced. And the very last thing is that we know with other gene therapies that have been done, sometimes if they integrate in the bad place, right, they go into the genome. If they integrate into the genome, if they go into a bad place, they can disrupt things. Now, there's been a lot of work to minimize that effect, but it's theoretically a possibility. So the very last part of it, in the last couple of moments, I just want to talk about, can we correct the gene mutation? This is kind of like the holy grail, right, of disease. Can we go in and just get rid of the bad part and put in the good parts? This is gene editing. So you're probably all very familiar with the CRISPR-Cas9 system. This is a system that comes out of bacteria. It's a protective system in bacteria that we've been now exploited in invertebrate biology. So the Cas9 is a protein that is able to cut and do double-stranded breaks in DNA. It has it recognizes a specific sequence. It's called a PAM site. And then we can target our CRISPR protein, a Cas9 protein, to a specific place using a gene-specific sequence. That's the sort of the sig RNA that you see there. So when this is activated, you get a double-strand break, and there's two ways this can get resolved. One is called non-homologous end junction repair. Problem with this is it introduces mutations. It's great to make mutations, but that's not what we're trying to do. We're trying to correct the mutations, so that's a bad outcome. It makes it maybe even worse. So that's what we're going to avoid. But there is another way that this will repair itself, and that's called homology-directed repair. And for homology-directed repair, you provide a template, and that template will have the sequence that's the correct sequence. And so it'll use that to then do the correction and repair of the mutant sequence, and it will actually introduce it. And so that is, in fact, an effective way to be able to really edit the gene. So the advantage is, of course, it fixes the root cause. You don't have to worry about gene regulation, and you don't have to worry about permanence. It's a solution. You get it in, you've done it once, one and done. The challenge is, we still have to avoid off-targeting events. There's still really limited experience with this in humans and whole organisms. It may not work well for large deletions or complex mutations. A single point mutation, that might be pretty easy. But if you have a multi-base pair deletion or some other complicated thing, that's going to be tough. Again, we're getting back to, just like the gene therapy problem, we still have to get all of that machinery into the right cells to do the change. And that's still a delivery challenge. Although I saw, again, somebody at the ASN presented an abstract claiming they were able to inject it via, I think, I'm not sure if they did a direct viral injection to the kidney or they did it through the ureter, but in either case, they claimed to be able to get pretty good activity. So we'll see. And then finally, because most PKD mutations are unique to the individual, personalized therapy is going to be required for a lot of people, and that can get time-consuming and can get really quite expensive. So it's the holy grail of corrective therapy, and there's massive progress going on in this space, but I still think it's going to still be a bit into the future. And so finally, I just want to close by saying, short term, I think most therapeutic options are still likely to come from treating altered downstream pathways. I think there's emerging insights that offer intermediate hopes of treatment that either replicate PC1, PC2 function, or can boost their levels or correct the effects of missense mutations. I think this is going to be the second level of effectivity that's coming soon. Again, I think there's hopefulness that we can reverse the disease if caught early. And again, I think true cures that are based on gene therapy or gene editing are going to still take some time as we work out the kinks. But I think, again, there really is quite rapid progress in this space. And so it comes back to us again as a community to think about how we're going to come up with careful and thoughtful clinical research designs that are going to allow us to, again, test all these different therapies effectively in our communities. With that, on time, exactly on time.
Thank you very much.
We can jump to
go ahead with your quiz.
Sure. So it's just two questions. The first one is a set of which of the following are true statements? One, our better management of individuals living with ADPKD over the past 30 years has significantly delayed onset of end-stage kidney disease in most. Two, the clinical presentation of ADPKD poses both opportunities and challenges for therapeutic development. 3, therapeutic choices may depend on the degrees of disease severity, an individual's age, and the tolerability of a treatment. 4, therapies directed at downstream pathways are more likely to be brought into practice in the short term. And 5, it is highly likely that pathways found upregulated in ADPKD tissue are contributing to the pathology of the disease, and treatments directed at blocking them will be effective. These are pretty easy questions, just saying. So, let's see here. They're still voting. So walk through quickly the answers on this. So our better management, number one, I would say that has not significantly delayed it yet. That was one of the first slides I showed. It was memory retention here. Could you remember for 30 minutes? So no, unfortunately, we haven't yet made it, although we're hoping that the tolvaptan will show us that. The clinical number two, the clinical presentation proposes both opportunities and challenges, and that's true. By the way, maybe I wasn't clear. Many of the statements could be true. There isn't just one true statement, just to be clear. Sorry? Yeah, just make sure. I wasn't probably clear enough on that. 1, 3, so 2 is true. Choices may depend on the degree of disease severity. I think that's true too, right? You can take something a little bit more risky with somebody who's got more severe disease than you might with somebody who's got very mild disease. Therapies directed downstream pathways are more likely to be practiced. I think tolvaptan is one. I think we're going to see others. So I think the answer to that is true as well. I mean, they may not be as effective, but they're going to almost certainly be more likely to come into practice, I think. And finally, number 5, is it likely that pathways found upregulated in tissue are contributing to the pathology of the disease? And I think that, again, most people got that right. That's not, I think, not, it often is true, but it's not a universal fact, and we can't assume that. One more question? Oh, we can skip it. We can, either way, time?
It's there.
Oh, okay. So which of the following are true statements regarding treatments directed at the root cause of ADPKD? 1, in most cases, mutations reduce PKD function, so therapies must be directed at improving or replacing gene function. By the way, more than one can be true or more than one can be false, just to be clear. 2, gene editing or gene therapy can provide a lasting cure. 3, we know that the disease is reversible if caught early, and therefore we can move ahead now with starting gene therapy clinical trials. 4, drugs that increase expression of either PKD1 and/or PKD2 may be effective treatments. 5, the primary obstacle to developing treatments underlying the underlying gene defect in ADPKD is due to the size of the PKD1 gene. And lastly, strategies used to treat cystic fibrosis may also be effective in some forms of ADPKD. So okay, we're waiting. Here we go. Voting. Okay. So I'd say in most cases, mutations reduce PKD function, and so therapies must be directed at improving or replacing PKD function. I think that's true. That's the whole 2-hit story and the haploinsufficiency story. Gene editing and gene therapy can provide a lasting effect, a lasting cure, if we're able to do it. We know that the disease is reversible. Can we go ahead with starting gene therapy? Well, we'd love to be able to, but unfortunately, we don't have the tools to do that, so that's a future state. Drugs that increase expression may be effective treatments. The answer is yes, that's true. They are in some cases. And then finally, the primary obstacle to developing treatments is the size of the gene. And this is a little bit of maybe a more complicated question. So in terms of gene therapy, it's true. The size, that's one of the barriers is the size of the gene. Delivery is still a second problem, but the size of the gene. But in fact, the primary obstacle to developing treatments underlying the gene defect is that we don't have technologies yet for getting effective delivery into the right target cells. And so I think that's a bigger obstacle. But this could go either way. Great. It's kind of like in Wait, No, Don't Tell Me. There's a radio show in the U.S. They often have a question where both answers are true. They just to make it for the audience. Okay, great. And finally, the last question. Strategies to treat cystic fibrosis. The answer is yes. Right? Protein folding correctors, those can also work for PKD. Presumably may work for some forms of PKD. So great. Thank you very much.
Thank you very much, Greg. And especially your talk is very illuminating in helping us to look into the crystal ball of what is yet to come, which I think is very important and inspiring for our patients.